cavities are no joke, right? If they get out of hand and they progress to the point of periodontal disease, even if they don't progress that point, bacteria can get into the bloodstream and can cause systemic infections, disease, and even death. It can cause sepsis. And so understanding a little bit about oral microbiology is essential. And what we're going to do is we're going to go ahead and tie in a few of the general components you've learned in the cell structure and function section, such as capsules, biofilms, fimbria, and adhesins, and apply it to dental caries. So what is a dental carie? A dental carie is essentially a cavity. And if we look at this series of images up here, what we're seeing is a cross-section of a tooth and gingival tissue. And what we see is we've got some plaque, which we've got biofilm in there. So the plaque starts and then it progresses over and to the point where then it has even entered where it can interact with the bloodstream. And so if we look at what we're looking at here, we're talking about production of biofilm from sugars, okay? And so bacteria, and the ones particularly in your mouth, such as Streptococcus mutans, Streptococcus salivarius, Streptococcus sanguinis, they take sugars and they assemble them, and they assemble them into long polymers called glucans, and which can create them with a capsule. So basically they're coated with this thick, sticky polysaccharide. Okay, and so part of a capsule allows them to evade the immune system. And so things like antibodies binding to sugar coating is not as effective if those antibodies should normally bind to a different cell structure. So capsules allow bacteria to evade the immune system. When enough bacteria are there and they're producing capsules and other glucans, basically you get this covering of a microbial community in this mainly sugar polysaccharide coat, but also a fair amount of proteins are mixed in there as well. So it basically creates this like three-dimensional world that they live in. And so that allows them to stick. Okay, so now they're stuck onto your tooth. They're metabolizing, and so for example, Streptococcus mutans can take sugars and ferment them into acids. And that acid gets to be high enough concentration that it can overcome the buffer in your saliva. So remember some chemistry 101. A buffer are chemicals that can bind and release protons, and so they maintain a pH around a certain range, right? So it's buffering capacity. Well, these bacteria can produce so much acid, for example, lactic acid, that they overcome that buffering capacity, and that acid actually then starts degrading the hydroxyapatite that is your tooth. So it'll break down the enamel and it'll break down the bone. Okay, so fermentation of sugars into acid by Streptococcus mutans is the main culprit for dental caries. Okay, so if we look down here then, this is to remind me to talk about lactate, and I've got another slide for that. So then periodontal disease is perpetrated by Porphyromonas gingivalis and Candida albicans, as well as others. Now, the thing about periodontal disease, it is around. So now we're talking about not just the tooth, but we're talking about is the area around it, okay? Ligaments, getting down into the bone, the tissue, okay? So then periodontal disease gets to be a whole nother problem as opposed to just a small cavity, okay? And so one of these players, Candida albicans, you've already heard about, right? So Candida albicans causes what? It was in the sexually transmitted diseases section, although it's not necessarily an STD. That's right, yeast infections. So the yeast is Candida albicans. And Candida albicans also causes thrush, right? So some of you may have seen it, maybe not, but that's where you have an overgrowth of this yeast in the mouth and the tongue, going back to the throat, and it's like this white goopiness. Really nasty stuff. Okay, so thrush, yeast infections, and periodontal disease can all be caused by Candida albicans. So if we look, let's go back to Streptococcus mutans, and remember Streptococcus mutans can produce lactic acid, And also, if you remember from Chemistry 101, that essentially, conjugate acids and bases, we have this where lactic acid can then dissociate into lactate and its proton, okay, and so decreasing the pH. This lactate can then be used by Candida albicans. So it's one of these crazy, not so crazy really, it's really ecology, where you've got Streptococcus mutans taken in sugars from what you're eating, convert it to lactic acid. The acid can drill into your teeth, and the acid, the lactate, can then be used by Candida albicans not only as an energy source, but also it's known to influence how Candida albicans actually grows. Okay, and so there's some evidence that can actually help it grow to its hyphal form, and the hyphal form is the form that is the infectious, more pathogenic one. Okay, and so to look at what we mean there, if we look here on this right-hand side, these images I got from this website down here, this is in its yeast form. This is a microscopic image, and these are just diagrams underneath it. This is in its yeast form. This is when it's starting to form pseudohyphae, and these are hyphae. So these, whenever you see Candida branching off, that means that it is getting more of the potential to be pathogenic. Okay, and that's what we're talking about, pathogenic Candida albicans, okay, periodontal disease. And so this paper that I'm having you read, right, you won't understand a lot of it, but use the directed questions and go through and understand what you can. Basically, Streptococcus mutans, Candida albicans, and the human mouth, a sticky situation. So a sticky situation, think about it, we're talking about sugars. If you spilled a sugar solution, right, and let it evaporate a little bit, it would be sticky, right? That's what we're talking about, and that is what, when we talk about glucans being formed in biofilms. Okay, so let's look at the images here from this paper that you're looking at for your reading. And so this is actually an extracted tooth, and if we blow up this region, we see this, and if we blow up the region even more, this is what we're looking at. Okay, so first I want to direct you to these white arrows, and unfortunately my arrow isn't working because of the program I'm using, but the white arrows here are showing channels. And so basically you can imagine this biofilm, which is what we're looking at here. It's like a sponge, right? And it's got pores all through it. And then think about that sponge being laden with yeasts and bacteria, hundreds and hundreds and hundreds of different species in there. That's what we're looking at. Okay, so right here, the water and nutrients can flow all through this. This is a biofilm. And what we're seeing now is these long hyphae. So this structure right here, right, is a pseudohyphae or a hyphae. And then we've got some yeast forms. And if we go back one slide, that's what we're talking about here, right? We're talking about the yeast forms, the pseudohyphae, and the hyphae form. And so now what we're looking at is a biofilm that's got that. And the blue arrows, I'm going to move the cursor out of the way, but the blue arrows are showing you bacteria that are associated with the Candida albicans. Okay, so then that is Streptococcus mutans. If we go to the next slide, what we're looking at is essentially discs of hydroxyapatite, which is mimicking teeth, right? A lot of our teeth is hydroxyapatite, as well as our bone. Okay, there's also a collagen protein coat. You may have learned about it in another class. But let's look at these images. And so we're looking at different magnifications. This right here is a hyphae of the Candida albicans, and then you see the bacteria, all of these little cells, right? And then here's another hyphae, and then here's Streptococcus all on it. Here's another hyphae, and here's a Diplococcus right there. You'll learn more about that next week. Singlecoccus, Streptococcus, and then these are the hyphae. And so what this is showing is an intimate association again in the kind of simulated tooth, right? But also what I think is fascinating about this image is what they're showing in the white arrows here, and also, although they're not showing the white arrows, if you look at this area, there are these holes in the hydroxyapatite, and the authors of this paper, the scientists, believe that those holes are actually from the acid that's being formed. So we're actually seeing kind of a cavity being formed on these fake teeth. Okay, so if we look at it even more, yeah, that's what this image is for. So holes, a hole right there, a hole right there. And these are what we're looking at for this mixed biofilm. So this short lecture is really just talking to get you going on this paper, Streptococcus mutans, Candida albicans. We've got acids, we've got lactate being used, we've got biofilms, which are formed from mainly sugar coating, a thick goopiness, and capsules, and bacteria will also attach by pili and fimbria, which are also in your readings. So don't forget to brush, don't forget to floss, and every once in a while you may want to use some mouthwash to help clean that out as well.